It's a late talk, so thank you very much for making it here. And today I'm going to tell you a little bit about deep learning approaches for the genetic analysis of high-dimensional phenotypic data with a focus on imaging datasets. So yeah, this slide tells a little bit about me, but Annalisa already went through the different stages. And one thing maybe that's worth mentioning, I'm switching back to academia from a position in industry. I was at Insidra for a few years, working on a combination of StatGen and machine learning for target prioritization. There was a drug discovery startup. And studied at Helmholtz Munich as a PI in January. And I'm part of the AI for Health Institute and almost at the Helmholtz Pioneer Campus. The group focuses on system genetics and machine learning. Okay, so in 15 years of genome-wide association studies, we have identified thousands of genetic variants associated with the complex traits and diseases. However, there's been great promises of the impact that these findings will have on personalized medicine and preventive care, but the actual impact has been relatively limited. And one of the main reasons this is the case, that we still lack a comprehensive understanding of which are the intermediate biological processes that are underlying the manifestation and the progression of a disease. So for this reason, we've been gathering more and more phenotypic data in genetic cohorts. And now we have a very rich spectrum of data sets from genetics to different layers of phenotypic data, spacing from cell traits, such as gene expression, organ level traits, which we can measure through medical imaging. Think about histopathology images or MRIs, and medical records. So biomarker such as blood test values, psychological tests, metabolites, and so on and so forth. Now, these rich data give us great opportunities. We have this cohort now in the sample sizes of the hundreds of thousands, or almost millions of individuals. And thanks to that, we have a great power to, thanks to the large sample size. And we have all these different layers. So we can perform integrative analysis across those to really understand which are the intermediate biological processes underlying disease state and progression. And we can also identify through epigenetics and deep phenotypes that are now available, which are the environmental drivers of human disease. However, opportunities always come along with challenges. We need scalable methods. Genetic data, they contain up to tens of millions of variants. And now we have cohorts in the order of magnitude with 10 to 5 individuals. We have also to correct robustly for confounding factors, because at those sample sizes, confounding factors such as population structure, family relatedness start playing a very important role. And we need robust correction for those. And not to mention that even the difficulty that one has integrating all these different layers. We can combine them in many different ways, and we need to be very precise on which are the critical questions that we want to make and what's the strategy for integration. And by themselves, if you think about imaging data, such as 3D medical imaging, these are very high dimensional. And even considering this data modality and wanting to perform a genetic analysis of that, that already presents many statistical and computational challenges. So today, I'm specifically going to focus on this last challenge, and will tell you two examples in which deep learning can help us analyze high-dimensional phenotypes. So I will start providing a brief introduction to genome-wide association studies, and then we'll discuss a convolutional neural network approach to analyze H&E stained liver biopsy images, and then unsupervised method for brain MRI. And just to clarify, the scope of these two analyses is still to enable genetic analysis of imaging traits.
So, okay, genome-wide associative studies. I think most of you probably already familiar with it. I'll go through very quickly. So the goal is to identify genetic variants associated with phenotypic variation, for example, disease risk. And so in a case control genome-wide associative study, one has cases, controls, and the methodology is very simple. One tests whether specific alleles tend to occur more often in one of the two groups, in cases or in controls. And this type of design is used mostly for complex traits, so traits that are influenced by many genetic and environmental factors, and it tends to focus on single nucleotide polymorphisms, SNPs, variation that happens at the single nucleotide level. And just to tell you a bit about the data, so we have a set of individuals. This can range anywhere from the thousands to the hundreds of thousands. And for these individuals, we have a phenotype of interest. And we also have genetic information across millions of genetic variants. And one approach that is used for quantitative trait is to consider a linear model with Gaussian noise, where one tests for association between the phenotype and each variant one by one using this model. And the outcome of these analyses is what people in the field refer to as summary stats. So for each variant, you get the p-value of association between the variant and the trait of interest. You get effect size of the variant, the beta, the regression weight. You get the standard error of the beta and genetic information such as the frequency of that allele. And so now it's very common in the field that you have this big cohort with hundreds of thousands of individuals and hundreds of traits, and you can define phenotypes from this clinical data and run GWAS, and then the results from the GWAS are made available in form of summary stats. And this, so in that sense, we can say that the association between genetics and disease through GWAS is relatively a simple computational. So we use a simple computational approach, and the outcome is readily available and accessible through summary statistics. So, okay, so what else? What else do we have? And as I mentioned before, the great opportunities here is in what's in the middle. So how we integrate, how we do the modeling, and how we integrate all the different data layers to define intermediate phenotypes, many times using machine learning and specifically deep learning, and then performing genetic analysis of these intermediate phenotypes using the results from GWAS available through summary stats and the results of genetic analysis of these intermediate phenotypes to really understand what's the likely causal direction between those and really unravel disease. Great, so that sort of sets the vision and the goal. And now I'm going to discuss the first example, an application of convolutional neural networks to H&E stained liver biopsy images. And that is for a study of non-alcoholic steatohepatitis. So non-alcoholic steatohepatitis, NASH, is chronic liver inflammation, which is caused by the buildup of fat in the liver. And the activity and this, of the inflammation and the severity of fibrosis is assessed by pathologists by looking at the H&E stained liver biopsy. And the pathologist assesses these 4 different processes: accumulation of fat in the liver, namely steatosis, inflammation, hepatic death. This is a specific type of death known as hepatocyte ballooning, and fibrosis. And as you can see, these scores are discrete. They capture the phenotype in a coarse way. And if you perform quantitative analysis, for example, genetic analysis of these scores, these tend to be underpowered because of this discrete characterization of disease. So we thought, okay, maybe we can apply deep learning directly on the imaging data
and trying to learn a more quantitative characterization of disease and try to perform downstream genetic analysis using these learned phenotypes. So we did that. We analyzed 5 clinical trials of NASH from Gilead. These contain histopathology images, genetics, RNA-seq, and clinical biomarkers at 2 different time points, baseline and 48 weeks after the trial. So our approach in step 1, we trained a convolutional neural network to predict pathology scores from the imaging data. And by doing that, the network learned to extract a continuous version of these scores. And then in the second step, we use these continuous scores in downstream analysis, specifically association with genetics and RNA-seq. A bit about the architecture that we used. So the main challenge worked with HNE-stained biopsy images. These images are huge. And we typically, people deal with these huge images by breaking them into tiles. And the other big challenge is there's a lot of heterogeneity within a slide. And so one really needs to model, give the flexibility to the model of focusing a specific part of the biopsy in order to be able to predict the pathology scores accurately. And we did that by implementing an attention-based pooling mechanism, which takes multiple tiles representations from the same biopsies and pulls them together with attention to make a biopsy level score, which then we use in downstream analysis. And the attention-based pooling mechanism was implemented in a way that the model could focus on specific parts of the biopsy for scoring a process like steatosis, but different parts of the biopsy to score other processes, such as ballooning or fibrosis. So we, first of all, assessed the ability of the model to predict adult sample. So we trained the model on about 4,000 slides and tested on a held out set of 460 slides from different patients at clinical centers. And these are the type of predictions that we get. On the y-axis, we had the continuous score that we learn out of sample. And on the x-axis, we have the pathology scores. And we expressed the agreement as a streamline correlation between predictions and the ground truth from the pathologist. And these values are in the range of cross-pathologist agreement. We also can go back and look at the weights of the attention mechanism. And specifically for each process, we can look at which tiles have been deemed important by the model for the scoring. And if we do that, we can see that the top tiles for different processes, they align very well with features that are selected by pathologists. And these are almost textbook images describing these different processes. We then assessed whether the continuous score have evidence of containing more information than the pathology scores. And we did that by trying to predict from the 2 versions of the scores, 60 NASH serum biomarkers, simply using linear regression. And as you can see, so here on the axis, we have the streamline correlation between the prediction from the model and the serum biomarker values. And you can see that the predictions are always better when using the continuous scores. And in some cases, we have a statistically significant results. Finally, we performed, this is really what we were interested in. We wanted to empower a genetic analysis of NASH progression in the clinical trial. And so we did that using the continuous scores. And when we do, we found 2 genome-wide significant loci and several suggestive loci, and then followed up with a series of post-GWAS analysis to break it down to specific genes, variants, and pathways. Importantly, though, when doing the same analysis using the pathology scores, we couldn't find any significant association. So this really shows the benefits of using deep learning in this case to just increase resolution of phenotypes. Okay, I will stop here because the next slide is I'm going to switch topic and discuss about an unsupervised analysis of brain MRI. So the principle is going to be the same, still on use of deep learning or high-dimensional phenotype for extraction of traits with downstream analysis. But we'll go from a supervised setting where we know which are the traits we're interested in to an unsupervised setting where we want to extract phenotype simply looking at variation in the population. And before getting there, I'll stop and take questions if there is any. Yes, no questions on this stage.
You can also post the question in the chat if you want, now or at the end of the talk. And okay, so I see one question from Sonia. Maybe you want to unmute yourself and ask. Or I can also read it. What is suggestive significance? TH.
Oh, suggestive significance. Okay, so sorry, just go there.
No problem. Yeah.
Suggestive significance is it depends a bit on the context. So the exact level can change. Suggestive means interesting enough that we do follow-up analysis on the genes. It's not quite genome-wide significant, but significant enough that we're going to look at those hits further.
Okay.
And sometimes, this is as low as 5 × 10⁻⁵. I think this is the classical definition. In this case, we use the stricter threshold, but this depends a bit also on the expected power, sample size, and yeah, it's application specific.
Okay. Florian?
Hi, Paolo. It's first of all very nice to see you. And I have a quick question. So regarding the post-GWAS analyses. So you said, okay, when you performed that, looking at the pathology-derived scores, like 0 to 6, you had no significant hit. So do you think that may have to do something with the likelihood? Because I mean, I guess, with the continuous scores, you can use nicely the Gaussian likelihood, and you can have like a very nice model that fits those continuous scores well. Whereas I assume for this one, when you had like the discrete scores, did you also use a Gaussian likelihood, or how did you model these discrete scores? Did you take the ordering into account? Or
Good question. We started with the Gaussian likelihood as well, and then in the model where we predict the scores, we actually use a binomial likelihood. And we tried the same thing also in genetic association study, just to be fair in the test. And yes, also, which is still an assumption, right? It's still an assumption to use a binomial likelihood in this case, but we tried something different in the book.
Okay, cool. Thank you.
Thanks.
Okay.
Great. Thanks.
We can continue.
Okay, so yeah, a second example, I'm going to present analysis of brain MRI data using unsupervised learning method. So yeah, these are 3D medical imaging. They're huge. They contain tens of millions of voxels. And the typical approach is to focus on specific regions in the brain and using segmentation, estimate region sizes of these different regions, and then performing univariate GWAS on these region sizes, looking at variation across individuals. And this works. An alternative people have also considered doing mass univariate association testing between each voxel in isolation, one by one, with genetics. Of course, that's severely underpowered because of the very large number of tests, definitely not ideal. But we thought maybe we can do something in the between and use unsupervised deep learning approaches
to learn a latent representation of brain, and then perform an association analysis of this latent representation to sort of bridge these two extreme approaches to the problem. So we really thought about what's the generative model of a medical image. And so we have latent phenotypes that mostly capture brain morphology. And these are affected both by covariates. These are non-genetic influences such as sex and age. And then we have genetic influences. And then we have a second step in this genetic process where from the morphology, we get to basically observe the high-dimensional phenotype, the image. And we model this first process using a linear mixed model. This is a standard model in genetics. If we consider a single latent dimension, just for simplicity, linear mixed model basically we model this as a Gaussian distribution with a mean, which is a linear function of the covariates, and then a covariance function that has two different components. One part capture similarity between individuals based on their genetics, and the second part is just IID. And this part is very much used in genetics to account for genetic similarity and the fact that we expect that individuals with similar genotypes, they also tend to share similar phenotypes, and that's made maybe due to different reasons. The second process is that we use the convolutional neural network, where a structured latent representation is unraveled into an image through a series of upsampling and convolutions. And this is sort of is going to be the decoding part, as we're going to see soon in the next slides. So this is the genetic process. And then we use the variational inference approach, very similar to the one that is used in the classical variational autoencoder to perform inference. And we end up with a generalization of the variational autoencoder, where basically we have an autoencoder structure architecture where images are encoded in the space and then back to the original space. And the loss is given by mainly two terms, plus a regression term. The first term is a reconstruction loss, where we want to minimize the loss of information between the reconstructions and the original data through the encoding decoding process. And then we have a second part in the loss, which is which represents the statistical model in the latent space. And specifically here is the negative log likelihood of a linear mixed model. Importantly, there's a difference between this model and the variational encoder, which this part of the loss is non-IID. And so the assumptions that are behind the applicability of mini-batch gradient descent break down. And so one needs to be smart about how to perform inference in this setting in order to be able to infer the parameter efficiently. So let's start the simple case. Let's consider the IID model. So let's not consider genetics, just condition the covariates, and make the very simple question. If we just use a slight variation of the variational encoder, do we find new hits by doing a GWAS-associated representations compared to standard approaches? And so we applied this framework to 1,800 brain MRI scans, for which for a subset of 1,500 individuals, we also have imputed genotype data. And we consider the deformation field as the phenotype of interest for this application. One way to understand the deformation field is basically the vector field that describes the transformation that one has to apply to the reference in order to obtain the sample, for example. So it's a representation of the data where all images, all voxels are now registered to the same atlas and tells us how to deform that atlas to get to the sample. And yes, we performed analysis independently for 24 different regions, and specifically considered a 64 by 64 by 64 hypercube around these 24 regions. So if we just do a GWAS using region size for these 24 regions, the only hit that we find is the ApoE gene. While if you use a multi-thread GWAS in this later representation, we can find additional hits, and only part of them are recovered using a representation obtained through linear dimensionality reduction, such as PCA. But this once we find these hits, we can go back to the raw image and visualize which are the phenotypes that are associated with these variants. But here the main focus is really, okay, yes, we can solve using a very short encoder in this setting is advantageous, but can we actually add genetics in and model genetic effects during the phenotype extraction step? And so we solved this problem in a technical contribution, the Gaussian process prior very short encoder, where we consider a structured, we generalize very short encoders to the case where the prior on the data space is non-IID, and we specifically focused on imaging datasets that contain representations of objects in different views. And so a later representations is now generated by an object and the view representations, and then we have the observational process that gives us the image. And by using variational inference, very similar to the case we've been analyzing before, we get to an autoencoding sort of framework where the prior in the latent space is a Gaussian with a covariance function, which is the product of an object covariance and the view covariance. So one example of application, we consider images of people faces in different poses, and we can break down the correlation between images into correlation between people and correlation between views, between poses, and use this model for doing out-of-sample predictions. We did something that is not necessarily possible using an IID model, such as a very short encoder. I'm gonna skip this application to MNIST. That was sort of a benchmark task. Yeah, just because that we then we're going now back to apply this model on brain MRI, specifically modeling the effect from 632 variants that are implicated with the psychological, degenerative, and neurological disorders through GWAS. So we model the effects of these variants during the phenotype extraction using this approach. And when we do that, we increase power in the stream analysis. These are preliminary results, but we find a new hit, which is very interesting. We are validating in the cohorts. With that, I'd like to acknowledge a few people for the first project of people at in situ, specifically Mikael Bereket, Matthew Albert, and Daphne Koller, and also people from Gilead Sciences and the clinical trial investigators involved in the trial. And for the second example, the Gaussian process variational encoder in this application to brain MRI, I'd like to thank people from MIT, MGH, Berkeley, and Microsoft Research, specifically Adrian Darka and Anna Bankov. And with that, I'd like to go back very briefly to this idea of using machine learning and doing the modeling in this interesting box here and extract intermediate phenotypes, and tell you a little bit about what the lab is working on in this big picture. So, of course, we are very interested in this part where we do deep learning for imaging genetics. For example, we have a project running on histopathology, genetic analysis of histopathology phenotypes. This is in GTEx, where we have histology, expression, and genetics across 54 different tissues. And so we're trying an unsupervised approach to learn histological phenotypes from variation between biopsies and linking it to genetics. And we just got access to UK Bank, where we have 40K brain MRIs heart MRIs, abdominal MRIs, and knee BXAs, and with matching genetics. And so, yeah, we'll be starting working with brain MRIs, and specifically looking at neurological disorders. We're also very interested in this part here, multivariate GWAS. In the second application, where we use the unsupervised approach, we were able to start a multivariate phenotype. And sometimes these multivariate phenotypes can be still quite high dimensional. And so this still requires method development in the statistical methodology space to be able to robustly assess for association with genetics. And finally, given the links that we know through summary stats between genetics and disease, and the links that we can learn between genetics and these intermediate phenotypes, we also work on multivariate randomization approaches that enable us to assess causality, the likely causal interactions between these intermediate and global phenotypes. With that, that's all from me, and I very much look forward to your questions. Thank you very much.